Hello everyone. For this week's lab, we were tasked to make our own fermented food. I chose to make yogurt. For a bit of a background about this fermented dessert, yogurt comes from a Turkish word that means tart thick milk. Its origin is not singular, but can be found in nearly every culture that has kept animals for milk. Scientists then began to study the bacteria that made yogurt. Soon after, they combined selected strains, direct set cultures, and made this commercially available. This allowed the companies to make the same yogurt with each batch. Yogurt making turned out to be simple. From the internet, I looked up a simple way on how to make homemade yogurt. First, I heated a pot of milk until it steamed. Then I let it cool down a bit. I added next a cup of yogurt bought from the store to act as a starter. I left it for about 24 hours in a warm place to ferment. Now I have my own yogurt. The direct set starters we have in our commercial yogurt have the bacteria Lactobacillus bulgaris and Streptococcus thermophilus. The yogurt starters containing live bacteria are added since they will ferment the milk. Since I do not have a freeze-dried starter, I opted to use the yogurt in the grocery stores. I can use this homemade yogurt again as a starter for the next few batches, but it can weaken over time and lose its consistency. The metabolic processes involved in yogurt making is called lactic acidic fermentation. Fermentation is the chemical breakdown of a substance by bacteria, yeast, or other microorganisms, typically involving no oxygen, making it anaerobic and the giving off of heat. Lactic acid fermentation occurs through the process of glycolysis, in which glucose is broken down into two pyruvate molecules. The pyruvate molecules are then used to produce two NAD molecules and two lactate molecules, which are the final products. The lactose in milk is converted to lactic acid that lowers the milk's pH. When pH drops below pH 5, methacylsecins, a hydrophobic protein, loses its tertiary structure due to the protonation of its amino acid residues. The denatured protein reassembles by interacting with other hydrophobic molecules, and this intermolecular interaction of secins creates a structure that allows for the semi-solid texture of yogurt. The production of lactic acid forms the basic structure and texture of yogurt. However, other molecules contribute to the taste of yogurt. These include acetylide, an important flavor substance in yogurt, and tyrosine, a product of proteolytic activity, but can cause bitterness when the concentration is above 0.5 milligrams per milliliter. Temperature greatly affects the turnout of the yogurt. Since the bacteria in my starter yogurt are thermophiles, they will love higher temperatures, ranging from 42 to 45 degrees Celsius. I can say that I succeeded in making my yogurt. This is my first time making it, and after learning so much about it, I'm guaranteed to have my own steady supply of it. I am interested to know how the yogurt would turn out if I used the flavored ones as my starter yogurt. I would also try to incubate it longer to check the difference in the taste.